and good evening, and thank you all for joining us today's webinar on solving the clinical, logistic, and societal hurdles for disruptive technologies in kidney transplantation, and also for connecting on Transplant Live, the ESOT e-learning platform. I am Maria Irene Bellini, a general transplant surgeon in Rome, and I'm also the scientific coordinator for Transplant Live. Our aim is to inspire, connect, and innovate the community with the latest innovation in transplantation. So please take the time to sign up and visit www.esottransplantlive.org and www.edo.esottransplantlive.org to participate and experience, in particular, the online resource developed for the transplantation learning journey, TLJ 2.0. This completely new online experience, consisting, as the name itself specifies, of a learning journey, aims to promote interaction and discussion towards a consensus, well beyond the face-to-face event. While we look forward to the online meeting that will be held from the 15th to the 17th of November, and that I warmly invite you to register, we also invite you to attend the pre-transplant webinar activities from the 7 workstreams. So please do join us and stay connected through the discussion boards. Workstream 1 is led by Martin Nazens and Stefan Schneeberg on a new surrogate endpoint in transplantation. They had already held a very successful webinar on the 6th of July to discuss the current status of xenotransplantation. In this second webinar of the series, the tremendous progress achieved by xenotransplantation will be used as a paradigm for initiating discussion on other futuristic transplants, how to design the right clinical trials, the patients and the EU Commission perspectives too. We will discuss this in detail with our expert panel, so please do participate and post your questions via the Q&A function. Also, please specify to whom this question is direct so that we will make sure that this will be addressed. And we will be delighted to hear from you later on during the dedicated Q&A session. So without any further delay, I leave now the floor to my co-chair, Martin Ogin, that is an associate professor at the section of nephrology and transplantation of the Department of Internal Medicine, Erasmus Medical Center in Rotterdam, and that is also a member of the Workstream 1 on new surrogate on kidney transplantation. Please, Martin, over to you.
Okay. So thank you very much for the introduction. So as Maria introduced me, I'm a biologist from the Erasmus Medical Center, and I will give you a little bit of introduction to the reasoning behind this webinar. So this webinar is part of the second of the first work stream of the TLJ, which has the name New Surrogate Endpoints in Transplantation. And the idea is that new technologies will lead to the development of new types of organs. And the question is, how are we dealing with those organs? How do we evaluate whether they're good organs or bad organs? Or how do we evaluate whether they're good enough to transplant? And that is what we're discussing in Workstream 1, which will be held in the TLJ 15th to 17th of November. And in preparation of this work stream, there's a series of webinars that are organized to think already in advance about these questions. The first webinar was held in July, and it was the name of the webinar was Prepping Kidney Transplants of the Future. And in a minute, I'll give you a little summary of what was discussed in this webinar for people that were not there, and also to refresh your memory a little bit if you were there. And then we're into the webinar of today, which is solving the clinical, logistic, and societal hurdles for disruptive technologies in kidney transplantation. So in the first webinar, I think, I hope everybody can see this now. So the main topics of webinar 1 were the following topics. So when will xenotransplantation be called a success? And then we took an example for transplantation of xeno hearts. How close are we to implementation of this technique? And what are the regulatory hurdles for xenotransplantation? And how do we select patients for clinical xenotransplant trials? And the panel that was discussing these questions were Emmanuel Kozzi, Richard Pearson, Bruno Reichert, and Linda Scobie, which we thank all very much for the contributions. And a little bit about the outcomes of what we discussed. So why xeno and other futuristic transplantation? So one of the reasons is that these technologies would allow us an unlimited supply of organs. and they would be available exactly at the time when they're needed. It's a great advantage of the conventional transplantation. And then the question, so how far are we? So, well, a lot of biological restrictions have been addressed, and their genetic modifications and novel drugs have driven progression. So we know now what are the highly immunogenic genes in pig organs, and we know how to cut them out. We know that there are viruses that are incorporated into the genome of pig, and we know how to cut them out. So there's a lot of development that is making this kind of technologies possible in the near future. And also there are some success stories from xenotransplantations from pig to primates who were able to survive for several months. And the question, which patients do we then enlist for these kind of trials? And then the idea is that probably difficult to transplant patients are first on the list, and perhaps you can think of hyperimmunized patients. And then the question that will be further discussed today is, does a patient want these type of organs? And regulatory pathways were discussed. So how do we classify xeno and other futuristic transplants? Are they all the same? Or is one category more risky to transplant perhaps than another one? And also, what do regulators want to know? So if we know what the regulators want to know, then people can adjust their methodology to create these organs and have the appropriate quality controls. And then when is a very difficult question. Well, there are some trials going on already with pig islet cells, which is the first start for organs. Still a lot needs to be finalized, and including safety issues. But progress is definitely being made. So then briefly on what are the transplants of the future before I hand over to the other presenters. So xenotransplants, we discussed. Another type of future organs, and that is maybe already very, very close, is the resuscitated organs. So organs that are maybe today not capable of functioning well in a transplant patient can be repaired in organ perfusion machines, such as you can see here on the right hand side for a lung. That would allow, that would increase the donor pool. We can also go a little bit more fundamental. So if an organ is completely malfunctioning, we can decellularize it. So take off all the cells that do not work anymore and recellularize it with healthy young cells. And here on the left-hand side, you can see an example of that. It's a paper from Danielle Leuning from a few years ago, and she decellularized a whole human kidney. And then she reseeded it with different types of human kidney cells. In the brown staining, you can see the areas where the kidney cells are growing and proliferating. And then maybe the ultimate future organ is the organ that we build from scratch. So pluripotent stem cells can differentiate into any type of cells. So can we make organs with multiple types of cells from a clump of undefined pluripotent stem cells? And here you can see an image on the right-hand side of a kidney organoid. You can see, if you look well, some tubular and glomerular structures, and in brown, the proximal tubular cells are stained. So this is an example of how new technologies are developing to make tissues that look like functional organs from pluripotent stem cells. Then today we will discuss cross-species kidney transplantation, and we will discuss how patients will be selected and how trials should be designed. We'll also hear about the patient voice. So what do patients think about these type of transplantations? Do they want them, or are they concerned? And also we will discuss the future of transplantation in a European funding scheme. So then I would like to introduce Rainer Oberbauer. He's a professor of internal medicine at the Medical University of Vienna. He will give a presentation on cross-species kidney transplantation, and will discuss the thoughts on patient selection and trial design. Please, Rainer.
Oh, I had to unmute myself. So here we go. Thank you very much, Martin. My task is to discuss xenotransplantation, specifically kidney xenotransplantations, and the thoughts on patient selection and how trials could be designed. So I have no conflicts of interest with regards to this talk. Let me give you some few words on how clinical research is regulated in the European Union and also in the US. So the sequence of clinical trials usually starts from 0 to 1 and is about safety and efficacy. So phase 0 and 1 trials are usually designed and applied to healthy volunteers, which clearly is not an option in kidney transplantation, but I will show later some examples of how this could be done. Efficacy is rather easy because we know from clinical allotransplantation at what parameters to look at. Now, safety certainly is a concern, specifically now in the COVID area. People are clearly afraid of transmissions of viruses or any other unforeseen consequences of xenotransplantation. So there's a tight set of regulations. The clinical trials, the approval of clinical trials, are the responsibility of each European Union member state, and I've give you the link to the EUTrain website below. The first in main trials, there's a specific category where guideline exists generated from the EMA, which is the European Medicines Agency. And there are two links below. The last update was from 2017. And what's specifically applicable to xenotransplantation are the guidelines for ATMPs, which stands for Advanced Therapy Medicinal Products. And again, there's a link below. Let me briefly start about 3 points that I think are very important in first in main trials, and I underscored it in red. So the first one is to demonstrate the relevance of any animal model in terms of clinical applicability. And so for xenotransplantation, I think, as Martin mentioned before, the probably best model is peak to non-human primate models, and there are some data on it, but there are some caveats, so to speak. The choice of the subject for first in human trials are important, and I will speak about that. And there are two very nice analogous trials in hematology where patients are selected for first in human trials, and I think we could use that as a template. And very importantly, not easy, are the stopping rules for the Data Safety Monitor Board, because we're speaking about few patients that are enrolled in such early trials. And then the question is when to stop. If one out of, for example, 5 xenotransplants would fail or would give some signs of significant safety issues, or is it 2 or 3 out of 5? Because you can't really do any statistics on that. And this needs thorough discussion. So stopping rules are very important. Now, the ATMPs, again, the Advanced Therapy Medicinal Products, fall into 3 categories. The first one is gene therapy, and I will give you an example on that. Nobody can see my mouse pointer. The second one is somatic cell therapy. And clearly, what's applicable to xenotransplantation is tissue engineered medicines. And let me go through the assigned trials or registered trials in EUTrain. So it's about 20 to 40 trials each year that are registered. So that's the latest paper I could find on that. And not unexpectedly, the cell-based therapeutic products in yellow are the most registered trials, and tissue-engineered products in greens are a little bit behind it. But that's what xenotransplantation would specifically of modified pig kidneys, for example, would be applicable to. Now, I think, as I said, there are two scenarios which would be good templates for kidney transplantation. The first one would be gene therapy, specifically in hemophilia A patients. Why? Because as in kidney transplantation, the patient would have two choices, either to get a transplant or to remain on dialysis. So it's not a life-saving transplant. And in hemophilia A, the patient also has two chances. The first one, to accept gene therapy, which is usually an adenovirus that's modified as a carrier of the recombinant factor VIII construct, which goes then to the liver cell and produces by the liver cell factor VIII. But it's of note that the phase 3 trial has been recently stopped because of decreasing efficacy over time. Or the other option would be, if the patient would be afraid, or also the regulators, to have continuously or daily or weekly infusions with either plasma-derived factor VIII or recombinant factor VIII infusions. So this would be one kind of model how it, where it was done. And the other one about patient selection are oncology trials. These are the poor patients where all the third-line therapy failed. And for example, the ALL for acute lymphatic leukemia, third-line therapy would be CAR T cells. Also for diffuse large B-cell non-Hodgkin lymphoma, these are the candidates which would be enrolled in CAR T cell trials. And similar to transplantation, those patients, because they have no more B cells and antibodies, have to undergo IgG infusions every month, kind of similar to immunosuppressive maintenance therapy after kidney transplantation. So I think advanced therapy medicinal products actually progressing, not in xenotransplantation, but in other fields. And there's a nice kind of parallel, I think. Now, how I think should the study design look like? So in phase 1 studies or 1-2 studies, one probably should select highly sensitized patients, and I put in parentheses educated, because you will see in a moment why this is, who are highly sensitized with, for example, virtual panel reactive antibodies again above 99%. And if those patients would accept the xenotransplant, certainly we need to do intensive immune monitoring. And there are some recent papers of how this can be done. And the paper just mentioned here also had to basically stop the xenotransplant to non-human primates because of severe infections after 130 days. Now, the phase 3 design would be, I think, rather easy, so to speak. On the other hand, it's very difficult to assure equipoise, which is the fundamental thing in randomized controlled trials, because you cannot say, you know, you either wait for an allograft or you have a xenotransplant now. So there's really a discussion that would require the inclusion of many other disciplines like ethicists or clinical trialists. Then probably the immunological disadvantaged with an expected very long waiting time. What's probably unrealistic is the non-inferiority study design because of the sample size and duration of follow-up, and we speak of several hundreds of such patients. Now, there are companies, and I just mentioned one of them because they're probably the most prominent after the science paper from George Church about these perf knockout pigs. The company was started and received, I think, about 100 millions of venture capital recently. And you see, even they, with this huge workforce and expertise, are not rather close to clinical trials. They're all still in preclinical phase with kidney transplantation. Now, there is some success in xenotransplantation, and you may know that worldwide so far, 38 patients have received xeno-islet transplants. So safety issues were not detected in these 38 patients, but efficacy was not achievable. None of those 38 patients, half of them are from New Zealand, the other from Argentina, were free of insulin even after multiple injections of these xenograft islets. The problem with this is the company mentioned is the long follow-up, because you have to monitor those patients for a long period of time, and some of the patients just no longer want to come to the study site, which is an issue, I think. Now, population attitudes towards xenotransplantation, and maybe Kevin afterwards can speak about that. There's a paper from Spain. They were interviewing Hispanic immigrants in the US about their attitudes towards xenotransplantation. And I just enlarged one of the tables, and you see, depending on their education, the agreement of xenotransplantation increases, but still, even in those with university level of education, only half of them are in favor, and almost more than half reject xenotransplantation. So it's not only a matter whether we can achieve it technically, it's also an educational thing. Now, what do I predict for the future? There's a nice paper by David Cooper about xenotransplantation, and he kind of cites or quotes three very prominent figures in transplantation. About 50 years ago, Sir Peter Medawar, the Nobel Prize laureate in 1960, said, we will have it in 15 years. Clearly, that did not happen. Now, 25 years ago, Sir Roy Kane said, xenotransplantation is just around the corner, but it may be a very long corner, and he was certainly right. And the most conservative interpretation of xenotransplantation and potential success comes from Norman Shumway, the doyen of heart transplantation in Stanford, who said, xenotransplantation is the future of transplantation, but always will be. So I hope we can prove them wrong and be more successful over the next 50 years than we were so far. So thank you very much for your attention, and I hand over to Kevin. Thank you very much.
Thank you very much, Rainer. This was really a pleasure for us to listen to your excellent insight from the clinical perspective. I would like now to introduce our next speaker, Kevin Fowler, the voice of the patient. He's from Chicago, Illinois in the US. So futuristic kidney transplantation opportunities, challenges, and the role of the patient voice. Kevin, please.
Thank you very much for the introduction, and thank you for the invitation. So what I'm going to talk about is just really three areas. It's just following from the previous presentation, just talk about the opportunities, where I see some of the challenges, and then how to utilize the patient voice through this hopefully new future for people living with kidney disease. These are my conflicts of interest. And, you know, if you look at the waiting list, I mean, right there it tells you the need for new technologies. So actually looking at old data, but looking at how the wait list for kidney transplants grew in the United States in one decade is really kind of breathtaking. So I would certainly say that there is a need for new technologies. There's no doubt about that. And if you look at in the United States, there's actually something that's been very positive back in 2019. The executive order was issued on advancing American kidney health. And within that advanced American kidney health, there were three aims. One of the aims is increasing access to kidney transplants, and specifically doubling the number of kidney transplants available for transplant by 2030. So from that standpoint, things are aligning pretty well with where our kidney care system is hopefully towards. And with that point in mind, I would also highlight the fact in the United States, the people who really shoulder the greatest burden of kidney disease failure are the African-American community. 36% of the people on dialysis in the United States are African-American, even though they only represent 13% of the US population. So clearly there's a strong need from several levels for new technologies. No doubt about that. But when you look at, though, I think some of the challenges, and I think I'll just speak for the United States, but the amount of misinformation that exists is really, from my standpoint, really kind of staggering where things have gone in our country. And I view that as a really a big challenge going forward, but I think there are ways to overcome that challenge. So that's one challenge that I see. And then as a secondary challenge that I see And this is looking at patient activation measure. So this is actually something that was developed by Dr. Judith Hibbard. And in next year in 2021, this will be a quality metric measuring person's level of engagement with their care. But if you look at kidney disease overall, and generally speaking, the activation within that community is not that strong. And that's why, in our country, the CMS is moving to make this a quality metric so people are more engaged with their healthcare. So I look at those as two of the challenges, the misinformation that's become so prolific in our country. And then I think overall kidney disease patients don't have the strongest levels of activation. And so then I think going forward, I'm going to draw upon, my experience working as a volunteer with the American Society of Nephrology, through the partnership that exists between the American Society of Nephrology and the FDA, and they formed the Kidney Health Initiative to advance innovation in the United States for kidney disease. And one of the things that was formed back in 2015, there was a lady, Celeste Castile-Lee, who really argued vociferously to ensure the patient voice was captured within the kidney health. And I encourage you to go online and learn more about it. But the whole role of the Patient and Family Partnership Council is to serve as an advisory council to the board of directors and other members of the Kidney Health Initiative. And it's not a token measure. It's something where our voice is very active. These are just some of the examples on ways that we collaborate in terms of providing input, patient input into different projects. And then there's also, we can also suggest projects that we'd like to see done. But this is an example. I talk about not having token behavior, but this is actually a meeting that our PFPC had with Secretary Azar. He's right across the table, and he was really one that was the advocate or had the vision for the Advanced American Kidney Health. And we met with him and we gave him our unvarnished truth on what we saw needed to be done in kidney disease. And quite frankly, it was one of the more enjoyable events in my life was actually to sit across from someone who had over $1 billion budget and say, this is what I like to see in the future of kidney disease. And it's amazing what may happen here in the United States. But I will just say this as my takeaway, I encourage you to look at the FDA patient-focused drug development as a template, is that I think that if you're going to work with the patient community, it has to be very intended, very intentional, meaning that you're engaging with the patient community early on in the development, that there's systematic engagement, it's not a one-off meeting, and that the patient's perception of risk benefit is captured, and many times that differs from regulatory agencies. I've seen that firsthand. But I think most importantly is that people who are going to be the end users are viewed as equal stakeholders in this process. And with that, that's all I have. So thank you.
So thank you very much, Kevin. It was very nice to hear the patient's voice all the way from the other side of the ocean. We will have some questions for you later at the end of this session. So we encourage people to post their questions in the Q&A button at the bottom of the screen. Then I would like to introduce Anna Pia Papagiorgio. I have to think a little bit before I pronounce it right. She's a policy officer from the European Commission, and we're very curious to hear about her views on the future organs. Thank you very much.
So thank you. I hope you can hear me well. I'll just share my screen and then we can start. So I hope you can see the presentation. I realize I've actually got quite a lot to cover, so I'll dive in quickly. So just to explain briefly for those who might not be so familiar with the European Research and Innovation Framework programs, we're currently in the 8th framework program called Horizon 2020, and it's running up until 2020 with a total budget of 80 billion euros. However, in the previous framework program, FP7, which was basically also a 7-year program, we funded about 60 projects to the tune of 349 million euros, all looking into organ transplantation. So I just want to emphasize here that these projects, so before I go into the sort of areas that we cover, the numbers here for H2020 are actually not complete. They go up to the mid 2019, so I'm sure that this number is actually a underrepresentation of the funds that have gone into supporting transplantation research. So overall, we have funded many different types of projects. These projects have a variety of different approaches. Some are looking into how to improve efficiency, either to personalized treatment, looking for biomarkers, looking at immune responses to either I would say cellular transplantations as well as organ transplantation. And a lot of research has been supported looking into alternatives. These are predominantly in calls that focus on regenerative medicine. They cover also xenotransplantation, organ preservation, artificial organs, organoids, anything you can imagine. Also health service research and public health. And in order to give you a little bit of a flavor of the types of projects that we've supported, I thought I'd dive a little bit deeper into a couple of them that illustrate, I think, the variety and the breadth that the funding framework program supports. So I will now try and move to the next slide. Okay, I'll skip this. This is just to illustrate that within the H2020 framework program, there are many different instruments that actually support health research. So please don't only look at the ERC or the collaborative research parts of the program, but really also look across the board, because you can find funding calls that you might not know exist. Right, we'll go straight to the first project. So this is an FP7 project that finished actually in 2018. And I actually really like this one because it's a consortium looking at organ preservation across Europe. It actually contained many different types of partners, academic partners, industry partners, non-profit partners, and actually the European Society of Organ Transplantation was one of the beneficiaries in this grant. So what is interesting here is that 3 large international multi-center clinical trials were performed and completed, one looking at the use of normothermic perfusion in liver transplantation. This trial was completed during the duration of the project, and actually the results were very favorable and positive, and this was in a publication, I think, in 2018 and featured also on the cover of Nature magazine. The other 2 trials that they performed were looking into preserving kidneys, so trying to use organs that normally would be discarded, and they were successful in recruiting the patient numbers that they wanted. Follow-up has been completed, and I believe that they are now looking into analyzing that data, and I hope that we will hear about the outcomes pretty soon. So aligned with this more clinical work, there were also experimental work programs in this grant, and they also created a biorepository of samples that actually are accessible to research groups outside of the consortium. So I think you can go to their website and put in an application for what you want to use their samples for, and they will consider sharing that. So I think this is a nice illustration of what this kind of collaborative project can actually do in terms of research. The next couple of projects that I will discuss are actually rather recent, so I can't tell you about what they've actually managed to achieve, but I kind of like the areas that they cover. The first one is EUTrain. It is a project that started at the beginning of 2019, and this one will actually develop a risk stratification and prognosis system for kidney transplantation patients. So the idea, I think, is here to provide a tool for clinicians that could actually help predict what kind of rejection patients could have over the 3, 5, 7, and 10 time point. And the aim here is to also actually test this system in a real life prospective randomized controlled trial. So I've put in here all the websites. You can go to the websites of each of the projects and find a lot more information than what I can give in this very short presentation. The second project that I'd like to show is actually a project which demonstrates a little bit the type of work that we support. We have a lot of projects also in FP7 and now in H2020, which look at providing a cell therapy in order to improve sort of patient outcome and to avoid graft versus host disease. So this is a very large scale and very ambitious project, which will create a platform, an off-the-shelf T regulatory platform for a variety of diseases, but the one that they will test is, of course, in organ transplantation. Here they will do a large component of preclinical work on or product optimization, I would say, using TRex, but gene editing approaches in order to modify those TRex in order to be more efficient and targeting once given to the patient. And they intend to perform 4 clinical trials. So this is a very, very ambitious project, but one that's, I think, very interesting to follow. I think this is the 4th project and the final one that I wanted to present. And this is a project that started at the beginning of 2020. So it's very short. It aims in 3 months to actually create a sort of ready to go artificial organ for end-stage liver failure. Here they will integrate stem cell-derived organoids into a vascularized scaffold, which will then go into some kind of bioreactor. Can't provide the scientific details. And this will then be tested in preclinical setting so that at the end of the project, they are ready to go into first-in-man trials. So this is a really exciting project. And it's, I think, one to also keep an eye out on. I wanted to I chose these projects really because I think they illustrate the range of what is possible, and that within these European projects, we really try to support all different approaches. So I think it demonstrates that we are not too scared of bold and futuristic types of research. But I do want to also mention that ethics is a very important part of the research that is undertaken. And we really believe that it's only by getting the ethics right that you really can achieve research excellence. So in parallel to the scientific evaluation that takes place of proposals when they come to us for funding, there's also an ethics appraisal that is done, and that's to make sure that the research activities that are proposed to be carried out under the framework program are actually in compliance with ethical principles. So the most important thing is that they comply with relevant and national EU and international legislation. And I've just picked up here a couple of the common ethical issues that we see all our projects actually face, which is when they involve children, patients, or very vulnerable populations. Projects have to be very explicit as to how they will deal with that. Human embryonic stem cells is a very sensitive subject in Europe. However, well, not only in Europe, but however, we do support research that uses human embryonic stem cells, though this is monitored and under very strict rules. So, but it is possible. And the final 2 elements that are very important for us are privacy and data protection issues. With the GDPR coming into force, a lot of projects have encountered what a change in such an important regulation means for them. But I think in the end, the type of research that is conducted is going to be more robust and more ethical. So, and the final part is, of course, research on animals and non-human primates. So we do monitor these projects not only in the beginning, but throughout the duration, and ensure that the higher standards are used. So this is in terms of what does happen in the projects, but also Horizon 2020 has supported many activities involved in forming the debate and engaging the public on these highly sensitive issues, such as genome editing, stem cells, human-animal hybrids, and also recently there was a call for research proposals on the ethics of organoids. So I think we do try and cover all the different elements that are important for our society. Now, I am not from the European Medicines Agency. I'm actually from the DG for Research and Innovation, but we do collaborate very closely with our colleagues from the EMA. And I just wanted to provide a couple of slides on the support that exists for the research community. And so the EMA is able to provide scientific advice and protocol assistance to researchers who are developing either a product or want to perform a clinical trial. Of course, the advice has to be prospective in nature. That might seem obvious, but we see very often that researchers reach out to regulatory authorities very late in the game, and that often comes at a cost, and also a lot of effort has already been put in that might need to be undone. So here I've just provided a whole list of things that the scientific advice and protocol assistance and the different types of support that exists at the EMA. Please go to the website and you can see what is available. There are also fees and fee reductions for small to medium enterprises, certain academic institutions, and in terms of eligible costs for Horizon 2020 beneficiaries, we accept costs for scientific advice. So here I just also wanted to point out the PRIME scheme, which is another scheme of the European Medicines Agency. It's here to, its primary aim is to enhance support for the development of medicines that target unmet medical needs. As you can see, there is a whole, it's kind of like a support school system that the EMA has designed. You apply, and if your application is deemed worthy of this PRIME scheme, they will help you in the whole development of that product. So as you can see here, there's a whole host of therapeutic areas that have been supported, and a lot of applications that have been rejected as well. But I see that transplantation features on there. So I think this could be very interesting for this community. And finally, I've talked about the past, I've talked about the present, and I just moved briefly on to the future. So Horizon Europe is the 9th framework program. It's intended to start next year in 2021 and will go on to 27. The Commission has proposed a 100 billion research and innovation budget, but this is currently being debated between Parliament and Council. But the aim of this program is really, again, to strengthen the EU scientific and technological base, to boost its capacity to innovate, and of course, the competitiveness and the job security that comes with that. So I think it's really, no one will argue that it's important to deliver on citizens' priority, and really health always comes up as a very important one. And I think the last 6 months have demonstrated how important that is even further. So we believe that the added value of such a program is all these elements. I think I've already probably spoken way too much, but I'll just quickly finish off. It's not going to be a revolution, it's an evolution to what we saw in Horizon 2020. So the structure remains the same. A lot of the things that that people have learned about the program will remain the same. However, I think the important feature, and here is the sort of proposed budget allocation, but I think the important feature to remember is that really it is designed to be an impact-driven program. So though we're in the process of drafting the topics and the calls, and I can't say more about that, I do want to emphasize that applicants need to keep that element of societal impact and tangible impact, actually, for society in mind when thinking about applications. So I think I will stop there. And the last thing that I will say is that though there is a partial agreement between Parliament and Council on this budget or on the proposal for Horizon Europe, there are talks of cutting the budget down from 100 billion, which is what we proposed, to 80 billion. So I hope that people will plead to their politicians and their parliamentarians to advocate for research and to ensure that budgets really are as big as they possibly can be. So thank you very much, and I hand over.
So thank you very much for this nice presentation. So there's some questions coming in via the Q&A, and we encourage people to send all their questions to via the Q&A button. You cannot speak, so people cannot raise their hands. So please type in your questions. I can start with the first question. I would like to actually ask myself to maybe both Rainer and Kevin. So Rainer showed that uneducated people at least are not too keen on receiving a xeno organ, and that increases maybe a little bit when people get more education. But what do you think what we should do with that? Do we need to educate people and inform them, maybe convince them to accept those type of organs? Or do we say people just need to make their choice, and if they don't want it, that's fine? So maybe both of you can comment on that.
Okay, maybe I start. I think we're not there yet, to be honest, and nobody really knows how far in the future potential success lies. So I think it's much too early to discuss things that eventually may never happen. Kevin, what's your point on that?
Yeah, I think my sense is that, I think what I would recommend, and I think this applies to anyone, is that, depending upon where you're at the stage, right, when you're moving into clinical trials, that there has been engagement with the community. But I don't think there's anything to be lost right now investing in understanding the community's insights into this and try to understand why they think the way they do. Because I think you're going to see differences by different types of kidney disease patients. People like myself with polycystic, we have a much different view based upon our family history versus someone with diabetic kidney disease. So that's where I would say is I think there's value in investing and understanding the population. There's no harm, no foul.
Yeah.
Well, I think the difficult part is that we cannot educate the patients because we don't know, what the risks and benefits would be. So it's just a very superficial discussion, but certainly patients should be involved anyway.
Yeah, and I think that's the part is that, rather than talk about how we should educate, first understand the community you're working with. That'd be my first suggestion.
So thank you very much. I would like to thank all the panelists. I think we had a really comprehensive overview from the clinician scientist perspective with Rainer, the patients and population perspective with Kevin, and the EU commissioning and the funding perspective as well. I know the COP projects quite well. On the one on machine perfusion, I was involved. So thank you very much, Anna-Pia. I think there is a question from Munib Syed, who is asking why only pig is used. Why not other animals for xenotransplant? Why not monkeys to human xenotransplant? Probably for Rainer.
That's a very good question. That's around for decades, I would say. Formerly, the FDA banned basically non-human primate transplantation because of safety concerns. As you know, for example, the Marburg virus or the Ebola virus can spread from monkey to humans. And given the current COVID situation, I think it's not an ideal era to discuss it. And the other thing is that it's very difficult to host and breed large non-human primates in the amount that pigs can be done. And there are so many genetically engineered pigs available, as I mentioned, from different companies, that I think the research there is much more advanced than in non-human primates. So I think it's basically a safety concern that stopped it so far.
Thank you. Yes, there was a question. Yeah. And from the patient perspective, Kevin, a question that is mine. Would you, if you think there would be a risk, what would you like to receive as an informed consent? There is now the process of informing the patients well ahead in clinic. What do you think should be the information that we as clinicians should provide?
I go back to what I had earlier, Dr. Belidi, is that I would just say is that, engage the community early on in terms of, understanding what you're working upon. I just think the earlier the better, because you're going to figure out what they need by engaging and having those relationships early on. I just see professionally that so many times people who are the end users are not engaged until the trial protocol has been set. So I just keep saying that, engage as early as you possibly can to have that relationship, to have the knowledge. It's going to be so much easier on the back end. But I keep saying that, and I sometimes feel like I'm just talking to myself, because it's just trying to get people to realize it's just a matter of listening, right? And viewing the group as a stakeholder. So.
There are other two questions. So one from Emanuele Cozzi for Dr. Papagiorgio. Do we expect that COVID-19 will impact negatively? I think Rainer has already mentioned this. And this is on the funding for transplantation research, again from Emanuele. Thanks very much, Emanuele, for joining us. Anna Pia, yes?
Yeah, so I guess the question is whether it will impact transplantation research or just research in general. I think in terms of research, I want to be an optimist. So I will say no, though that is just what I wish. I think in terms of output of research projects, that what we see, the COVID pandemic has really been quite has negatively impacted the work that has been carried out. A lot of laboratories across Europe have had to stop the research that they were doing. Sending of samples and materials across Europe ceased for a couple of months, and it was only starting to sort of start up again, and now we're seeing a lot of restrictions again across Europe. So I think that this pandemic has a negative impact on so many things in our society, and of course research is part of that. But I do think that in people's minds, health has become an important element again, and therefore I hope that politicians will realize how important citizens value health, and research is part of that. So we will see.
Thank you very much. And then again, once for Rainer from Emilio Canovai, what are the remaining immunological hurdles to clinical implementation of xenotransplantation?
Well, there are recent papers from the Boston group, but also from Japan, showing that the old world monkeys are less than ideal for transplantation because some of them have natural antibodies against the triple knockouts or transgenic pigs, and so therefore much has been accomplished. All the perf versus has been knocked out, as I said, by the Boston group. All the gal knockouts exist, but by introducing, usually they use CRISPR-Cas, some of them using TALEN, introduce new genetic modifications that eventually identified as immunogenic, and some of the actually non-human primates showed preformed antibodies against those knockouts. So I think it's still a long way to go. By introducing 50 or 100 knockouts with some transgenes, I think it's a little bit uncertain what the whole genome looks like.
Thank you very much.
Thank you. So maybe I have a question before we're waiting for some other questions. So maybe this is, well, maybe to the whole panel, but maybe specifically to Anna Pia. So we've been seeing maybe that Europe is a bit more careful in implementing new technologies than, for instance, China or the United States. If we look at genetically modified crops, for instance, that are not really favorable in Europe, but they are grown on quite a large scale on the other side of the oceans. How do you think that will compare to the development of new types of organs? Are we going to be more careful than other countries, or you don't see that happening?
Well, that's a really difficult question, actually. But are we more technology adverse than other regions in the world? I'm not so sure. I think if we don't look at the historic and also things that happened, I think in terms of the genetically modified organisms discussion, there were lots of things that were at play at that time. But I think it sort of links a little bit to what Kevin was saying. I think you have to engage the patients and the public in these debates early on, and make them less futuristic, actually, and more accessible and more realistic, and provide people with the sort of what the opportunities lies in these approaches. So I don't think Europe is hesitant to embrace technology. I just think that we are perhaps not as good as certain other countries and other areas in having a public debate on these things. So I think that that's an area where we in Europe can definitely work on.
Yeah.
I was just going to add to this. I think there's a way for both the U.S. and Europe to work on this together. Because I don't think we're any more advanced than you are. That's just one person's opinion. But maybe there's an opportunity for both groups to work together. I guess that would be my hope.
Thank you. Time for one last question, probably. What's the future of TRex to produce tolerance in a human against xenotransplant? I think again for Rainer.
I get all the difficult questions. I think nobody knows, because even TRex and allotransplantation are somehow heavily studied, but there's no clear picture. I mean, there are some success stories on regulation and probe tolerance or tolerance protocols. In terms of xenotransplantation, I think there's not a single study that I'm aware of showing that there's any benefits.
Thank you very much. Again, I would like to thank all our panelists because it has been an exceptional hour with a very insightful and really up-to-date futuristic overview of what the future of kidney transplant will be like. There are other questions coming. I'm afraid that we might not be able to answer them live, but please do post them on our discussion board. There is a dedicated discussion board for workstream 1, so we'll make sure that everything will be everything will be addressed and answered. I would like also to encourage the registration to our online scientific session that will be held from the 15th to the 17th of November, where particularly on this workstream 1 there will be really plenty to talk. So please do join us. Remember ESOT TLJ 2.0. I would like, before leaving you, to ask you to please comment on this webinar. There will be a poll that will be launched soon. So please do stay with us and give us your feedback. And of course, if you have any recommendation for how we could improve future webinars, please leave your suggestions in the chat or email askme@esot.org. And there will be plenty of other webinars before the TLJ, so stay please tuned. And remember the learning journey that will not stop with the online meeting. There will be other webinars as well, and other questions that are coming as well. So, great. I want also to remember you that this webinar is being recorded and will be available within 3 days via the ESOT YouTube channel or in the TLJ ESOT.org program, the website, and the Transplant Live platform, with a full list of all the activities that are available for the 7 work streams too.
So thanks, Maria Irene and all the panelists. It was a pleasure, and have a nice evening.
Thank you.
Thank you.